So can we start?
Yes, please.
Success is not built in a single day. It is built every evening when you choose not to give up on yourself. Rest today, rise stronger tomorrow. On behalf of entire Agri SAARTHI team, it is my immense pleasure and privilege to warmly welcome you all to today's insightful session on the topic biofortification and maize breeding. It is rightly said, the health of a nation begins with the health of its food. In a country like India, where nutrition security is as important as food security, biofortification has emerged as a powerful scientific solution to combat hidden hunger. Today, we are truly honored to have with us our esteemed speaker and distinguished personality, Dr. Ramesh Kumar, principal scientist in genetics and plant breeding, and a renowned maize breeder. With decades of dedicated research experience in maize improvement, quality protein maize, and stress tolerance breeding, Sir has significantly contributed to the development of improved maize hybrids and varieties that strengthen both farmer prosperity and nutritional security. As Norman Borlaug once said, food is the moral right of all who are born into this world. Biofortified maize is not just a crop innovation, it is a step to our ensuring better health, better livelihoods, and a stronger future. Sir, your expertise in maize breeding and biofortification is an inspiration for young researchers and students like us. We are confident that today's session will enlighten us about the scientific advancements, challenges, and future scope in this crucial field. I would also like to extend our heartfelt gratitude to Dr. Opi Chaudhary, President of Agri SAARTHI, for his constant guidance and visionary leadership, and to Mr. Ajay Kasana, Chief Secretary of Agri SAARTHI, for his continuous support and coordination in making such knowledge enriching sessions possible. Agriculture is our wisest pursuit because will and contribute most to real wealth and good morals and happiness. Today's discussion truly reflects this spirit. Once again, I warmly welcome our esteemed guests, respected dignitaries, and all participants. Thank you. I now request Dr. Ramesh Kumar to kindly deliver his valuable address.
Okay, thank you for a long introduction. And I'm not a guest. I'm part of this Agri SAARTHI program, society, and I welcome all students who have joined this program. Today, I will deliver a small, you can say, in brief lecture, but I need one more day to teach the students this breeding. And I will try to have a board with me. I will ask my son to arrange a tab for me so that I can write with pen. Without blackboard, I cannot teach. It's my problem because while teaching, I need blackboard. So we will start just dear students, in this presentation, I have to cover two parts: maize breeding and biofortification and maize, with advanced tools or with conventional breeding methods. So basically, I was busy somewhere else because from tomorrow I have to go for a long tour for 10 days. I was preparing that myself for that tour. So I could not spare too much time so that I can prepare a special lecture on breeding. But I need a day, 2 hours to teach you breeding. Maize breeding means all complete breeding. every breeding method is developed on maize only. So we will start from heterosis breeding. Heterosis breeding, what is heterosis breeding? Heterosis breeding means, you can say directly hybrid breeding also. When you cross two parents, the F1 will be superior to both of the parents. It is not only superior to the mid value, but it is superior to the highest value of the both parents. And there are different systems, that dominance, overdominance theories, that I cannot tell you in detail today. That's why I need one day, complete 2-hour class to teach all these things. So in heterosis breeding, what I have told, that what is heterosis? Heterosis is superiority of F1 over both the parents. And in case of maize, we usually CGMS lines, and these are not required at all. However, we are using that also. We are developing lines for CMS system lines also in maize also, particularly in baby corn, so that we can avoid detasseling. But in case of field corn, we used to do detassel because the detasseling is quite cheaper than to spend money on that aspect. And long back, there was one issue that at that time the CMS lines were used to produce hybrid seed. But due to outbreak of one disease, North Corn Leaf Blight disease, during that time, it devastatedly was there, and whole germplasm was lost. That's why we have avoided the CMS systems in maize and started normal detasseling for pollination. It is very easy also. So crossing the inbred lines, indigenous, exotic, that is a different thing. I have already told that crossing of two parents, you will get F1, and F1 is superior to both of the parents. That is single cross hybrid. And during this lecture, I will inform you what is single cross hybrid, what is double cross, what is three-way cross hybrid, how we are developing that. Hybrid varieties or hybrids first time commercially exploited in maize only. Long back, in 1878, Beale reported that certain varietal crosses he has developed, and he got heterosis up to 52% or more than 50%. He was very happy to see that what happens. Then now all of you have studied that when the term heterosis was coined by Shull has given this term in 1909. Double crosses, Jones in 1918. And different, this is the top cross hybrid. Then who has given these methods? So prediction methods are, Jenkins has provided prediction method of double cross performances. This prediction of performances, that needs a complete lecture. Few years back, I was teaching hybrid breeding course in Punjab Agricultural University. Though I was not employee of PAU, I was employee of ICR, but voluntarily they have given that course to me. I have taught that course for a long. So I need complete lecture on one aspect. Then in 1944, about 80% of the area that was covered by maize hybrids. The total area under maize cultivation at that time, it was covered by hybrids only. But there were some issues that come down again. The hybrid performance was not altogether better at that time. In America, the inbred lines were poor. Then they sit together and discuss what to do. Then they decided that till our inbred lines are not so strong, we will not do hybrid breeding. That they have done double cross hybrids. They left the single cross hybrid, moved to double cross hybrids, and till the time they have improved their inbred lines, they have not touched the single cross hybrids. Once they got confidence that now our inbred lines are perfect, then they again put single cross hybrids in market. And right now, as on date, USA is leading in case of corn production and productivity, and their average productivity is 11 ton per hectare. That is a miracle. Though they have single crop in 1 year, it's a almost 1 year crop or 10-month crop there. So due to temperate environment, there is an added advantage. In India, it is not possible because we have a very hot and humid environment. During Kharif season, basically our maize crop in India is rain-fed crop. So it matures only in 3 months. In 3 months, if your hybrid is giving 5 ton, 6 ton, 7 ton yield, that is far, far better than 10 months crop. So 1960 single crosses have become widely accepted. After improving their inbred lines, they again launched the single cross hybrids and they were very popular. In India also, in 1952, this program was launched. And a number of inbred lines and germplasm were introduced from US and India, but that were not suitable to India. They were not adapted at that time. They have taken some time. Though at that time they have released few hybrids, Ganga 1, Ganga, Ranjit, Dakkan. They were the earlier hybrid. They have released and put in Indian market. They have provided to the Indian farmer, but it was rejected after some time because our farmers were not so strong financially. They can purchase hybrid seed at that time, and they need only the seed which we can use time and again, year after year. So in India, our scientists, our seniors, they discussed, and again they followed the same American policy that we will develop first our germplasm. Till then, we will provide the composites and synthetics to the farmer so that they can save their own seed and cultivate without spending too much money. And after that, after some time, you can say our former director, Dr. Sai Das, he took charge in 2006. Then he made a policy that we will not allow any company or any public sector institution to contribute a double cross or 3V cross hybrid to the ICRP system for testing, and we will not allow them to sell in market. So after 2006, you can say the real hybrid era starts in India. And during last 15, 16 years, we have crossed approximately, we have increased our production 3 times. And every year our production is increasing. So there are some demerits also. Merits. What are the merits? सिंगल क्रॉस हाइब्रिड में सबसे अच्छी बात यह कि सब पौधों की हाइट एक जैसी होती है, सभी का एक ही जगह कोब लगता है, सबसे ज्यादा यील्ड देने वाला हाइब्रिड सिंगल क्रॉस हाइब्रिड होता है. सिंगल क्रॉस हाइब्रिड इज द मोस्ट extraordinary potential, you can say, among all these hybrids: single cross, double cross, 3V cross, top cross, synthetic, composite, etc., etc. Among all these, single cross hybrid is the highest yielding hybrid. Crop will be uniform. Genetic constitution of all the whole crop will be same. It suits mechanical harvesting. For mechanization also, that is very much suitable. You can take up sowing with machine. You can do hoeing operation with machine. You can harvest with machine and put in dryer to dry the grain and sell in open market. So these are some benefits. And what is the demerit? Foremost demerit is farmers have to purchase new hybrid seed every year, because they cannot use the next generation of a hybrid crop as a seed. That will become F2, and you know, in F2, this will segregate. 50% will be hybrid, and 50% will go towards the parental lines. The crop will not be uniform. There will be no uniformity at maturity stage also. Some mature first and some mature later at later stage. So mechanical harvesting will not be possible. So that's why we cannot advise a farmer to grow F2 seed in his field. Second point is hybrid seed production requires considerable technical skill. Yes, technical skill is required. When you put your crop, male and female, together in field, you need to detassel the female line at a particular time. Within 2-3 days, you have to detassel the whole female crop, female plants, so that the female line, female part of that female line, that is known as silk or stigma, will receive the pollen from the male parent, and the seed which will be set at female line will be hybrid seed. Very simple. You no need to put pollen by hand. Once you detassel female line, there will be no pollen from female parent. Only female part is there, and the pollen will come from the male parent. This will be available in abundant quantity, and pollination will be done through wind only. हवा में ही उड़ेगा और अपने आप पोलन गिरेगा, सीड बन जाएगा. Exploitation of full potential of hybrid varieties requires an adequate supply of irrigation, water, fertilizer, and control of weeds, disease, insect. Yes, hybrid crop always needs more fertilizer. They are input responsive. Their potential is very high. That's why you need to give more. जैसे पहलवान है, पहलवान की खुराक अलग होती है, तो हाइब्रिड इज पहलवान. Simple words. The large scale production of hybrid seeds depend on easy emasculation. No need of emasculation. It's very easy, no problem. Every farmer cannot be ready because the seed yield will be less. And hybrid crop will give 10 ton yield, whereas the female parent or hybrid seed production, that will give only 3 to 4 ton. So there is too much gap. Farmers sometimes they shy. They think that they will get loss, but the cost of hybrid seed will be more. That's why he will not be in loss. Always the farmer who is producing seeds for any company or any public sector organization, he will be benefited. Never he will be in loss, but people think so. So different types of hybrid. Single cross hybrid, then modified single cross hybrid, double cross hybrid. What is single cross? That I have already explained, that crossing to diverse parent, you will get F1. That is known as single cross hybrid. Simple, very simple. Only 2 parent you require. And for single cross, you need only 2 isolation. For maintenance of these lines, you need only 2 isolation. One for female line, one for male line, and in one field you will make cross to have hybrid seed. What is modified single cross? Sometimes what happens, the female line, that is somewhat low production line, low productivity is there. Or you want to increase the quantity of hybrid seed. What is the method? Then what you will do, while breeding, you will have so many inbred lines. From one source, you will develop so many lines. But in these lines, there will be minor differences. For example, tassel gloom color is different, or silk color is different. Only minor difference, but there will be no difference in yielding ability. So what we do, that we will cross 2 sister lines first to exploit the heterosis among these lines, between these lines. We will make a cross, and then that seed will be used as female seed. Again, plant that and then cross with B or male parent. So this we used to call as modified single cross. Though it's a clear-cut three-way cross, but the female sister lines, two sister lines, there was not much difference. Only one gene difference was there, or two gene difference was there. Slight variation in character. That's why they have some variation. To exploit that variation, we can produce more seeds by crossing to the male parent. That is modified single cross hybrid. Then double cross hybrid. In this, we will make two crosses. A into B, F1 आ गया. Next one, C into D. It means 4 inbred lines are involved. A into B, then C into D, and दोनों का जो क्रॉस होगा उन दोनों क्रॉस को मिला के हम हाइब्रिड बनाएंगे by involving 4 parents. That is known as double cross hybrid. In case of three-way cross hybrid, that I already explained in case of modified single cross, one F1 is crossed with third parent. A inbred line, B inbred line, we have made one cross. Then this F1 is crossed with third parent. It means 3 lines are involved to produce the three-way cross hybrid. In double cross also we can modify this also. However, in case of double cross, modification is not required at all because already we are having 2 F1 crosses. So there will be no dearth of seed quantity. That's why usually we don't go for double cross, but some people, some companies, they are doing modification in this case also. Though right now we are not allowing them to put in AICRP system or to sell in the market. But as you know that Indian market is open. We cannot control. We cannot go everywhere. So there are chances that people are doing that also. Modified three-way cross again. You can see in this. A into B, one F1 was there, then it was crossed with C. But they have some variation in C also. They are crossing that C with its sister line C1, then make crosses. This is the modified three-way cross. Then top cross, it's very simple. One inbred line is crossed with open pollinated variety. Open pollinated variety means synthetics or composite, which is having broad, you can call that is a broad-based tester. Means many parents are involved in this. Open pollinated variety means there is open pollination is there. So many parents are involved. From that diverse source, we can cross inbred line that is known as top cross hybrid. Sometimes this method, usually top cross hybrid, we use to see the combining ability of the inbred line. For that only we are using this, otherwise we are avoiding this. We are not doing any top cross hybrid, developing any top cross hybrid to give to the farmers. Then you can see this single cross hybrids with picture also. This is inbred line A, that is known as female parent or seed parent. We will do detasseling in this case, and the pollen parent or inbred line B, that pollen will go to the female part of the inbred line A, and the seed will be hybrid seed. So commercially, what we are doing that 3:1 ratio, 1:1 ratio, 2:1 ratio means we are growing 3 female line and 1 male line. Or some companies, they are doing 5:1 also, 6:1 also. It all depends on weather conditions. If your environment is humid and hot, that you can go for 5:1 also. If your environment is somewhat dry, then don't take any risk. 3:1 is the best ratio for that reason. Similarly, in case of double cross also, you can see with this picture, 2 parents, we will cross them to have one F1. We will cross these 2 parents, we will have one F1, then we will again cross both these, and we will have the double cross. In case of double cross, what we will do, 8:1 or 10:2 means there is too much pollen or line is productive. When you made cross for C into D, means F1 is there and F1 is always having higher pollen producing ability. So there will be no dearth of pollen. You can go for 10:2 or 10:3 or 8:10. You can increase the lines of female lines. Three-way again, I have already explained this also. And in this case, the male parent will be inbred line only. So pollen availability as compared to the double cross, in this case again with inbred line, less pollen will be there. So 3:1, 4:1, 5:1 is enough. By this, you can produce good hybrid seed. Then we are talking about inbred line. Inbred line, female A, B, C, D. I have spoken 4 types of inbred lines. What is inbred line? A true breeding line obtained by continuous inbreeding or selfing. That is called as inbred line. We used to self the line or sib mating, you can say, but sib mating will take more time to achieve the highest level of homozygosity, whereas in case of selfing, it will be achieved in less time. Inbred line consists of individuals with the same genotype. When the line is fixed, F6 or F7, you know that more than 90% homozygosity will be there. So all plants in case of one line will be similar. Then inbred lines are developed from a variable source population. Why? Variable source population means we need strong inbred line, we need diverse inbred lines. From one variable source, you can develop hundreds of lines. If your population is not variable, source is not variable, your all inbred lines will be same, look like alikes. Then what will happen? Heterosis will not be there. If your source is diverse, your inbred lines are strong, they are productive, they are diverse, then there are more chances of having a very good and productive hybrid. So what will be the source population? Source population must be an open pollinated variety or synthetic. That is the most preferred. But we used to develop inbred lines from single cross hybrid or double cross hybrid also. Double cross is also having a broad-based as compared to the single cross. Because in case of single cross, only 2 inbred lines are involved. In case of double cross, 4 inbreds were there. So the population will be diverse something. But in case of open pollinated or synthetics, you have involved 10, 20 inbred lines. If 10 lines are there, then population is automatically will be diverse. So to develop any inbred line from a variable source is most important. Then inbred line development, how we are developing that? A breeding method, you all know that pedigree, backcross, single seed descent method, there are different breeding methods. But most preferred is pedigree method in this case. However, we are using backcross also. In some cases, single seed descent also some people used. But pedigree and backcross, these are the 2 major breeding methods by which we are developing our inbred lines. What is pedigree method? You all know. 2 diverse parent, we will cross them, we will have F1. Then we will self that F1 to have F2. And from F2, we will select plants, desirable plants with desirable traits as per our objective. Then the single row progeny will be grown from those plants, and we will select the desirable plants within that progenies. And after reaching F6, F7, the line will reach to the highest level of homozygosity, and that is known as inbred line. And then we will evaluate for a particular trait for which we have developed. If the inbred line was developed for long cob, then we will select the plants or progenies with having long cob, or if it was developed for thick cob, then we will select thick cob only, or seed weight, or you can say that this bold kernels, etc., etc. For a particular trait for which we have developed the inbred line, we will select that only and reject all other material. After each self-healing desirable plants are selected, that I have already explained, selected and self-pollinated. पीपीटी में यही दिक्कत है कि ये चीज जो मैं आपको अगली लाइन में पहले बोल जाता हूं, यदि मेरे पास ब्लैक बोर्ड और चौक होता तो मैं आपको करके दिखाता हाथ से कैसे करते हैं, आप कभी नहीं बोलते उसमें से. Then the inbred lines, developed inbred lines, they will be evaluated for their per se performance, means yielding ability. उनकी क्या एबिलिटी है उसके लिए हम उनको परफॉर्मेंस चेक करेंगे. परफॉर्मेंस चेक करके उनमें से बेस्ट को छांटेंगे. फिर देन वी विल कैटेगराइज देम इनटू टू डिफरेंट ग्रुप्स, फीमेल एंड मेल. उसके लिए एक डिफरेंट मेथड है. कैसे हो सकता है उसको करके दो कैटेगरी बनाएंगे. फीमेल के पेरेंट क्या होते हैं कि फीमेल का पेरेंट दैट शुड बी प्रोडक्टिव, दैट शुड बी स्ट्रॉन्ग, two most root system should be strong, lodging should not be there. These traits are for female parent, and male parent always should be high, more height. And most important trait in case of male parent is pollen producing ability for longer duration. The male parent should produce pollen for 1 week continuously. If the pollen is drying within 1 or 2 days, we cannot use that line as a male parent. We need minimum for 1 week. So there is some strategy also how to develop inbred lines. There are some strategies. Strategy में short term and long term. We are following 2 types. Short term means immediately we need inbred line. So there is only one method, pedigree method. You can cross 2 parent, diverse parent. You can have F1, then self F2, and you can reach to F7. The lines will be fixed. Or in between, you can test these lines F3 onwards for early generation testing for disease resistance or insect resistance or any other trait you can test. Discard those lines which are not having desired traits. But in case of long-term strategy, it's a really long-term. You cannot develop inbred lines in 2-3 years. It will take long time. In last slide, what I was discussing that we need a diverse source. So in this case, we will develop diverse source first. For example, you have 100 inbred lines, 100. Or you have parental lines of 10 hybrids, means 20 lines. Then first what you will do, we will generate information of heterotic grouping. We will divide all these lines into 2 distinct groups, female and male. Then from female group, for example, you have 10 lines that are behaving like female lines and that are good lines. So we will make chain cross. Number 1 line will be crossed with 2, number 2 will be 3, 3 with 4, 4 with 5, 5 with 6, 6 with 7, 7 with 8, 8 with 9, 9 with 10, and 10 with 1. Each and every line will get chance to cross with each and other line. Then all these crosses we will have some seeds from every cob. From middle of the cob, we will take 20-30 seeds from each cross. Then we will mix all and then plant in one field. Then allow to random mate. We will grow that mixed seed in isolation so that pollen from outside will not come there, and that each and every plant will get pollen from that field only. So that will complete one cycle. Next year also, after 2-3 cycle, we will start selecting plants from that population. Then we will self that plant, those plants, as per our objective. And when, for example, from that population, we have selected 50 plants with low cob placement, long cob, medium height plant. We have seen these traits are available in these 50 plants. Then from those 50 plants, we will grow year to row progeny. In first generation, you have selected 50 plants and self them first time. Then we will grow progeny of each plant. It means 50 lines we will grow next time. And in that individual row, we will see which plant is not behaving like others. We will select maximum plants which are similar, and then mix the equal quantity of seeds from these plants and grow next generation. Like that, in every year we will grow. So by reaching F6 or F7, you will be having fixed lines. So it will take 2 more years, because first we have categorized the inbred lines into male and female. It will took 2 seasons. Then after categorization, you have made chain crosses, 3rd season. Then chain crosses के बाद में आपका real शुरू हुआ है, though, so it will take 5-6 more seasons. So approximately you need 10 seasons to develop inbred lines by following the long-term strategy. Whereas in case of short-term strategy, it was only 6 to 7 seasons. So 3-4 seasons extra we need. That's why it is known as long-term strategy. So after fixing these lines in both types, once the lines achieved F7 level or more than 95% homozygosity level, then we will evaluate them for their performance, per se performance, combining ability, and categorize into distinct groups, and then made available them to a new breeder to make heterotic crosses to have good hybrids. So these are the strategy to develop inbred lines. Then in addition to this was the conventional method. DS technology is also there. What is DS technology? Haploids. You all know better than me, because you have already read these to appear in the MSc examination. An individual with the gametic chromosome number in its somatic cells. And you need to use colchicine. You will give colchicine treatment to that haploid to double that chromosome number. That is known as haploid to double haploid. We will double that. So that is double haploid DH lines. In maize, how we will develop? This is the conventional method which I have explained to you in last half an hour. And in case of this DH lines, what we will do? This is the source population. Source population may be inbred line, maybe F1, maybe F2 segregating population, or maybe a composite or synthetic. That is all our source population. What we will do? We will make crosses with inducer. What is inducer? Inducer that will create haploidy. by using conventional method, we will make cross. The first applied maize was developed by Stadler and Randolph. Spontaneous applied. It was selected under field condition, spontaneous mutation. So induction rate was very less, only 0.1%. When they see it and used it, it was, you can say it was only study purpose, 0.1%. Then one more scientist, he has developed that stock 6. It was having 2.3% induction rate. And slowly, right now, we have inducers which has the capacity of induction of more than 15% right now under Indian condition. So again, we have achieved this. This is a remarkable achievement by our colleagues. So this is the method which I have already explained, but once you made crosses with inducer, and inducer, it has a morphological marker system for identification of haploids. This blue color will be there, violet color will be there, and you can see this is hybrid. In case of hybrid, your whole kernel, embryo as well as the top of the grain or seed will be colored. Some light color or dark color will be there. But in case of haploid, you can say the kernel with the haploid embryo have a significantly lower oil, as well as it has only the top color is colored, otherwise it is not colored. So we will select these. Once we have made cross with inducer, then we will have to select the grain or seed with this color only. Then we will grow. We will be able to know that yes, this is the real plant, and then we will give colchicine treatment. After that, it will be, the chromosome will be double, that is known as double applied. So by using this method, within 1.5 years, you will have very good inbred lines. But we need mild climate for this program. Under harsh climate, we cannot do that. Otherwise, because haploid plants are, but in case, so we need a stable environment without any variation in the environment. So our country is a big country. We are lucky to have each and every environment at one time. We are having cold in Jammu and Kashmir or northern India. The temperature is going below 0 degrees C, but at the same time in southern India, the temperature will not go below 10 degrees C. That is the most favorable environment for maize breeding. That's why all private companies, they are working in southern India, either in Maharashtra, Karnataka, Telangana, Andhra, they are working there because that environment is stress-free environment. During winter season, there is no rain, there is no pollen possibility, pollen will be available, they have irrigation facility, there will be no pollen mortality, there will be no heat shock, there will be no drought. So 100% congenial, optimum environment is there. That's why the private companies have gone there. They have selected that area to have more hybrid seed, or seed producibility is more. Our breeding program is very much successful there. In northern India, we are having extreme environments. During winter season, the temperature will go below 0, and during summer season, or you can have the temperature more than 46, 47, 48. At that time also, it is not possible to do maize breeding. So both types. I am sorry to say that the place where I am sitting or where we are working, it is not suitable for breeding. But in our country, Policymakers are different people. They are taking decision as their wish. So now we will come to the malnutrition issue. So it is almost 1 hour that I have spent on maize breeding, but too much I have left. I have to explain, I wish to explain each and everything in maize breeding. Very slowly, what is introduction, then how we are doing pedigree crosses, what we are selecting, what we are rejecting, that needs to be elaborated at length. Then we will go to the recurrent selection methods, particularly recurrent selection methods. Which method we are following, that needs to be explained, but we will take it at a different time. So in between, I wish to give 5 minutes to discuss these breeding methods. If any students wish to ask anything up to this level, I am ready for that. If anyone wish to ask anything? Okay. I have to stop here for some time. Students, yes. 5 minutes, 5 questions you can discuss with me. This is an interactive session. First, this 00444 Aspharia. Aspharia. Then Kanishka Yadav. Unmute yourself and ask what you want to ask. Kanishka. You cannot unmute? Organizer, they can unmute the student who want to ask any question. For 5 minutes, we can discuss breeding.
Okay, thank you, Dr. Sir. I would like to call upon Parveen Kumar 05894. Please unmute and ask your query.
Hello, am I audible?
Yeah, audible.
Yes, sir. Thank you. Thank you for the great session you gave us on breeding program, sir. I have a doubt here. I have been working with maize seed production companies,
Okay.
but what I feel personally is that nowadays, instead of breeding, going for a hybrid seed, can we not promote varietal seeds for maize? What is your opinion? What are the advantages and disadvantages of that?
I will not advocate for varietal seed production varieties because
What would be the reason, sir?
India needs more and more maize, and the production capability of varieties is not so high. F1,
Can we
I
not improve
already
that one?
explained that F1 is more potential than any other. So why we will go for less yielding?
Okay, but can we not improve the performance of varietal seeds? What do you think, sir?
We can improve, but we cannot improve too much. We cannot improve beyond hybrids. Once we have improved our inbred lines, that inbred line will be used to develop that composite. Rather than going for composite, we can use that inbred lines for producing F1. And F1 will be more productive then. If your inbred lines are productive, then F1 will always be productive.
All right. A same question, a similar question related to this. Is it possible to have a variety which is better resistant than a hybrid in our case? Like, a maize hybrid which is having susceptibility or disease susceptibility. Can we not have a variety of that kind which is tolerant to the disease or pest?
Variety has a buffering capacity as compared to hybrid, more buffering capacity. But hybrid is always strong. They can avoid, they can escape the disease. They can tolerate the disease. if the inbred lines are developed in such a way that they are already tolerant. By using tolerant inbred lines for making new combination, and that hybrid will always be tolerant. So in every case, hybrid is far, far better than any variety or composite.
Thank you, sir. Thank you. Thank you very much.
Next one.
Okay, Yadra Minaji. Okay, Ayan Goda, please ask a query. 03956, please unmute and ask a question.
Ayan Gowda. Good afternoon. Ask
Sir,
directly. No need to good afternoon or good evening right now. We don't
sir,
have
among all the hybrid seeds, which one is better? The single hybrid seed or double hybrid seed? Or
single cross. Only one answer, single cross.
yes, sir. I have another question, sir.
Yes.
In recent years, hybrid seeds are not majorly used for the cultivation for the farmers, which does not come for the next generation after sowing.
But in India, area under hybrid is increasing every year. Right now, throughout country, it is approximately 80%. And when I started maize breeding, I joined 15 years back this institute. That was approximately 50%. And in these 15 years, this situation has changed a lot. Only those areas or during Kharif season, some area is there, particularly in Bihar or you can say in hilly region, they are not sure that crop will mature or not because flood situation is there. That's why they are using composite or F2 seed. Otherwise, in Rabi season, that area, they are using 100% hybrid seed. They know the potentiality of hybrids. Perhaps
Okay,
I
sir.
will answer your question.
Okay, thank you, sir. Now I would like to call upon Ms. Tata Sai Monica. Please unmute and ask your query.
Namaste, sir. I'm Sai Monica. I have a question. If we keep on doing producing plants more tolerant to pest and diseases, is there problem of rising of super pest? Do we get that problem in the future?
No.
No. Okay, sir.
It is not genetically modified. We are not
Okay, sir.
producing any chemical. We are doing it genetically.
Okay, yeah.
Conventional methods. We are
That mean
developing.
it's genetically modified. We are producing genetically modified plants,
There will be
right,
no
sir?
outbreak like that.
Okay, sir.
We have different populations. Every
Okay,
hybrid
sir.
has a different genetic background.
Okay, sir. Then these plants comes under genetically modified plants, right, sir?
No.
No? Oh, okay, sir. Yeah, okay, sir. Thank you.
Next one.
Sir, on the behalf
But your voice is not clear. I am not able to hear.
Okay, Simran, please hold for a moment. We can take some more
आप एक काम
questions.
करो, आप वीडियो बंद करके बोलो, आएगी आवाज.
डॉक्टर साहब अभी एक दो क्वेश्चन और ले लेते हैं, उसके बाद अपन नेक्स्ट उस पे चलेंगे. V Vandini 04442, please unmute and ask your query. V Nandini.
Yes, good evening, sir. Myself, Nandini. I'm from Agriculture BSc, Hyderabad. I have a question
BSc
that
horticulture.
Agriculture, sir, BSc Agriculture.
Okay. Which university? PJTSU?
PJTU, sir.
Okay.
Yes, sir.
Yeah.
Sir, I'm really interested in GPPR. Does there is a scope future in GPPR?
Yes, scope. You want to ask scope? Breeding is the best subject.
Sir, what are the books we need to refer?
You
What
have
are the
plenty
books we
of
need to
jobs.
refer?
Number of jobs are available. You can join any seed company. You can start your own entrepreneurship in seed production, or you can be a seed certification officer, or you have chance to become one agriculture officer in production program, or in general also you can go. Most important thing you can join any country. This depends upon your potential to achieve the highest level in science. Everywhere breeders are required. आप
And sir,
मोलिकुलर काम
another
कर सकते हो
question.
ब्रीडिंग में, आप कन्वेंशनल काम कर सकते हो. You have number of opportunities.
Okay, sir. Thank you, sir. And
Scope is
another question
quite
is,
wide. Yes.
and another question is, what are the books we need to refer for MSc and JRF exams in genetics and plant breeding?
Nandini, you need first basic book. Then for genetics की बुक आती है. This is very basic book.
बीडी सिंह की आती है सर.
नहीं, बीडी सिंह I will not advocate for that. बेंजामिन का आता है, एक बुक आती है, बेंजामिन राइटर है. दैट इज वेरी कंसेप्चुअल बुक. और ब्रीडिंग में आप एक बार पढ़ोगे तो बीडी सिंह की ठीक है, बट बीडी सिंह को पढ़ने के बाद दूसरी बुक जरूर देखें. बीडी सिंह सिंपल, बहुत सिंपल है. बहुत ज्यादा सिंपल से काम नहीं चलता है. आपको जेनेटिक्स की डिफरेंट बुक लेनी पड़ेगी, ब्रीडिंग की डिफरेंट लेनी पड़ेगी, तब आप कवर कर पाओगे. कोई नहीं, इसके ऊपर कभी अलग से ब्रीडिंग का कोई ऐसा सेशन रखेंगे, उसमें डिस्कस करेंगे. कोई कुछ पूछना चाहे तो, नहीं तो फिर हम नेक्स्ट शिफ्ट करते हैं.
एक लास्ट क्वेश्चन पूछेंगे डॉक्टर साहब. बाकी एक जो करियर गाइडेंस और करियर काउंसलिंग सेशन अपने 30 दिनों में होगा, तो बच्चे उस समय जो पूछेंगे तो ये सारे सवालों का जवाब मिल जाएगा. तो आदित्य कुमार जी का क्वेश्चन है एक. गुड इवनिंग सर, माय क्वेश्चन इज व्हाइल क्वालिटी प्रोटीन मेज एंड प्रो विटामिन ए एनरिच्ड hybrids have been successfully released, what are the current challenges in staking multiple nutritional traits like iron, zinc, and vitamin A into a single multi-biofortified hybrid without compromising yield?
Dear, we are working that. I am program leader of this program since last so many years, and right now we have developed genotypes with high lysine, tryptophan, provitamin A, low phytic acid, vitamin E. All these traits we have stacked in one genotype. For iron, zinc, we are working on that also. We have first taken up zinc, and we have found quite variability also, and we have exploited that and developed inbred lines with higher zinc content. But iron is very tricky. Frankly speaking, to work on iron, it is very difficult. Iron is changing once you have any contamination at harvest level. Once you have harvested the crop, by chance your cob is put on soil, land in field. It comes in touch with the soil. In soil, lot of iron is there. What you have bred, but it will be changed while estimation. In lab also, what equipment or what container you are using in which you have stored. It is made up of iron, then again iron will be more. It will be inflated. Even you are making chapati, when you put that raw chapati on your tawa, it will have more iron. It will suck from that also. So at every step, iron is variable. So it's very difficult to estimate. It's very difficult to quantify that. That's why, but we are studying the correlation, and iron is positively correlated with zinc. That's why we are working on zinc. Once we have increased the zinc, iron will automatically be increased. Or we have lowered down the phytic acid. Once phytic acid is up to a level that is reduced level, 30-40% than the normal genotype. And the availability of iron and zinc will be more for the consumer. So by that way, we are addressing this challenge, and all these traits which you have asked, we already stacked in same genotype. So in coming days, perhaps this year during workshop, we will be able to release one high iron zinc hybrid also. Anyone else? अजय, मुझे तो लगता है कि आज के सेशन में हम ब्रीडिंग तक सीमित रखें, ज्यादा बेटर है। बायोफोर्टिफिकेशन को आज छोड़ दें, क्योंकि दो चीजें मिक्स होंगी, फिर दिमाग में कुछ और जाएगा। और किसी को 10 मिनट हम और डिस्कस कर सकते हैं, ब्रीडिंग वी कैन डिस्कस।
तो देख लीजिए डॉक्टर साहब, अगर क्वेश्चंस लें तो बच्चे तो बहुत सारे क्वेश्चंस पूछेंगे।
नहीं,
और
मैं
अगर
ब्रीडिंग
कंटिन्यू
के
कर
क्वेश्चन, मुझे क्वेश्चन आंसर करने में क्या प्रॉब्लम है? अच्छी बात है,
थोड़ा
बच्चे
सा
पूछें।
15-20 मिनट का थोड़ा सा ओवरव्यू इनको दे देते बायोफोर्टिफिकेशन, क्योंकि वो भी
चलो
बहुत
फिर
जरूरी
मैं
है।
नेक्स्ट स्लाइड लगाता हूँ,
हाँ
ठीक है?
जी, हाँ जी, उसके बाद क्वेश्चंस
Okay,
ले लें।
then I am sharing again, and I will try to complete that in few minutes.
Dear participants, you can note down your queries and there will be ample time provided to you to address your
So,
queries.
dear students, now we will touch this malnutrition issue or nutritional aspects. As you know that throughout world, the malnutrition problem is there. More than 2 billion people, they are suffering from malnutrition issues, micronutrient malnutrition, and approximately this 750 or 800 million people, they are undernourished. When we talk about this children of less than 5 years, 150 millions are stunted, their growth is not normal. Then more than 45 million, they are wasted. You can say their waist is not developed well, or approximately 40 million, they are overweight. And 11% of GDP in Asia and Africa that goes waste to solve these issues. We are spending too much money to address these issues, and it is very difficult. So a huge amount of your GDP is going towards that direction. If it is already solved, then your 11% GDP is a huge amount that can contribute to the national development. Under Indian situation also, the same is the situation. Out of 640 district, 240 district, they are having stunting levels more than 40%. It means half of the India is suffering from malnutrition. It varies from state to state, area to area. Somewhere it is 10%, somewhere is 70%. So this is a huge population suffering from malnutrition. Government of India has conducted one survey in 2015-16. On these 15 points, I have marked this neonatal mortality rate, like infant mortality rate or child less than 5 years mortality rate. But by pumping money in different schemes, either it is midday meal or infant food supplied in Anganwadis or different schemes to provide free this anemia medicines or iron pills to pregnant women, or this at early stage treatment to the newly born child. All these issues have been addressed by Government of India. And the second survey after 5 years, 2019 to 21, the situation was somewhat improved. The green color points, you can see, for example, at number 3, child less than 5 years mortality rate per 1,000 births, from 52 it came down to 40. So after every 1,000 we have saved 10 lives. So like that, the situation was somewhat improved, but in some cases it is still alarming, or it is increasing, like obesity, overweight, anemic conditions. So in this case, we failed to address these issues, and again government has put more money or started new schemes to address this, and the government has given a fixed some sustainable development goals that we used to call SDGs. And out of these 17 goals, 12 are revolving around these malnutrition issues. Different schemes for gender equality, good health and well-being, zero hunger, no poverty, reduced inequalities, clean water, sanitation, quality education, all these are addressing malnutrition only. So what happened? When we spend $1 or 1 rupee, you can say in simple term, you can say 1 rupee on nutritional program, you will get benefit of 15, 16 times you will get benefit. It means once our population is malnutrition free, they will be healthy. We can save our money, which we used to spend to provide medicines to improve their health, and that population will always contribute towards good. They will have better education, they will have better health, they contribute more towards country development. And our Honorable Prime Minister always used to say that young India, we have maximum young people in our country. Yes, it is true. We have more young people than any other country. And if the youngsters are channelized in a proper way, then they can contribute a lot to the country development. To address these issues, the government is providing iron pills. One my dear student has asked that whether you are working or how you will address these challenges of iron zinc. Yes, government is providing iron pills, but as a breeder, as an agriculture scientist, we are not advocating anyone to take iron pills. Instead of taking iron pills, they should take better food which is already rich in iron and zinc, or any other micronutrient which is deficient in your diet. Because once you take medicine, your bowel movement, पेट का जो समस्या है, आपको एक नया इशू आ जाएगा कॉन्स्टिपेशन का. आपका पेट क्लियर नहीं होगा, आप यू विल हैव मेनी डिजीजेस. आयरन की गोली खाने से आपका आयरन तो बढ़ जाएगा शरीर में, आपका ब्लड लेवल तो ठीक हो जाएगा, लेकिन जो बाउल मूवमेंट है, पेट की जो कंडीशन, वो खराब हो जाएगी. इससे अच्छा है you should take the nutritious food. So what we are doing at our institute to have maize with high lysine and tryptophan, that is known as quality protein maize, to increase the methionine content, to increase the provitamin content, to address the eye issue, to improve your eyesight, low phytic acid reduction program, so that we can made available the iron, zinc, calcium, magnesium, etc., all the micronutrients. And nowadays, Vexicon is also required with high amylopectin content, and that is used in food industry, or you can synthesize, you can have more ethanol content also. So biofortification, you all know that nutrient dense staple crop. By using the best conventional method and biotechnological tools without sacrificing any agronomic performance, and that trait should be of consumer preferred trait. So you can see this lysine and tryptophan we are discussing. In case of field corn or normal maize or non-QPM, you can say Out of 100 gram, it was only 1.3 gram in case of endosperm protein. Whereas in case of quality protein maize, it is approximately 3 times, 3 to 4 gram per 100 gram of endosperm protein. Lysine is 30 to 40% more than the field corn, and tryptophan is more than 50% than the field corn. We have developed these genotypes. So many hybrids are available. Biological value, in case of normal maize, it was around 40% only, whereas in case of quality protein maize, the biological value is 80%. Biological value means the protein which is available in your grain, your body is consuming that, digesting that up to 80% in case of quality protein maize. Whereas in case of normal maize, you can digest only 40%. 40% will go waste in your excreta. Protein intake utilization, you can see this. So less grain or less quantity of quality protein maize can supplement the required level of lysine tryptophan as compared to the normal maize. You will have to take 20 gram, 24 gram, whereas in case of quality protein maize, only 8 gram. So higher level of lysine tryptophan, and most important is biological value. Then two leading organizations, World Health Organization as well as the Institute of Medicine, they have calculated the what is the requirement of lysine and tryptophan, and how it is available, and how much we need to take, how much maize we need to take in our daily food to meet these requirements. In case of normal maize, you can say more maize you need to take to supplement that particular amino acid, where in case of QPM, it is less is required. Too much less is required, which is possible to eat. कोई बोले कि आपको आधा किलो मेज रोज खानी है, you cannot eat. और कोई कहे कि 50 ग्राम खाना, तो आप खा सकते हैं. कुछ अमाउंट मेज से मिलेगा. The rest amount of these amino acids you can take with other sources also, by consuming milk and egg, etc. But your major diet, your staple diet is cereals. From cereal you are getting, and maize is the only cereal which can provide these amino acids. So this is the, you can say, the middle one. The light cannot pass the black grain in photo, but this is not black. This is, you can say, this opaqueness is there. Light is not passing through this grain. That is opaque, and opaque is somewhat soft, and it invites insects, and the storability is poor. So what we have done, we have improved this by selecting these grains with less opaqueness. The area, opaqueness area is less, and the rest is hard kernel through which the light is passed with the required level of quality, tryptophan and lysine. We have developed this. So you can see the some children, they are having their health issues, and the last one is healthy one. To make our population healthy like this, we need such product which is rich in lysine, tryptophan, methionine, provitamin, etc. So low cost quality protein is available in the case of quality protein maize, and the quality is as comparable to milk. In simple terms, you can say one chapati of QPM will be as good as one glass of milk. It is meeting the energy and protein requirements. Then one most important example, in case of pig, 3.5 times faster growth is in case of QPM than the normal maize. The small one, you can say, At one stage, both were at same age, same size. But one is fed with normal maize, one is fed with QPM. And QPM fed pig is grow 3.5 times faster than that other one. In case of poultry also, the similar is the situation. With less quantity of grain, in less time, it will be ready to sell in market. It will be developed fully. More meat will be there. Same is the situation. QPM-based diet fed to the pig and it gained 256 gram per day weight. While in case of another one which is fed normal maize, it is gained only 21 gram per day. There is a huge difference. So lysine and tryptophan, they're playing a major role in developing the muscles. So right now, all of you, as me also, we love to have gym every day. And people used to have protein powder also. So if the protein powder is made up of quality protein maize and provided to the population, it is healthier, it is safest product, and you will have more muscle mass. So there are various benefits of QPM. High biological value, high lysine tryptophan content, health benefits, food safety, better climate response, wider adaptability, all you will find with this genotype. Because under field condition, you cannot differentiate this is QPM or non-QPM. Plant is same. Only difference will be at the time of maturation of the grain. The protein content is, ये क्या username, this is requesting remote control of your computer? No. So this is a normal crop with added advantage of increased lysine and tryptophan content. Food processing does not affect quality, QPM quality. When you made chapati and cook, the lysine tryptophan quantity will not be reduced. This is it. In case of vegetables, every doctor, every nutritionist, they will advise you not to boil it, not to cook in cooker. You can take it raw, but in case of QPM, quality protein maize, none will ask you to use in such a way. You can take it directly. You can roast the cob. You can eat. You can make flour. You can make chapati, halwa, puri, sweet, everything here you can make with QPM flour. It will be healthier. It will always have the same quantity of lysine tryptophan as it was in raw grain. So I am happy to share that Honorable Prime Minister has dedicated 17 biofortified crop varieties on the day of World Food Day, and out of that 17, these 3 hybrids belong to our institute. That was the first function organized to dedicate these biofortified crop varieties, and all these 3 belong to my breeding program group. Then one boy was asking that multi-trait stacking, multi-gene stacking. In this program, we have stacked protein, lysine, tryptophan, provitamin A, low fatty acid, all these in common genotype, one genotype. You can see the line as serial number 12. Lysine, the threshold level is 2.5. It is approximately 3.5. Tryptophan level, the threshold level is 0.7. It is more than 0.8. Provitamin A, that is threshold level is 5. It is more than 8. And fatty acid, that is 2.5 threshold level, and it is less than that, 2.06. So we have developed these lines, this material, this germplasm, by stacking all these traits, by following conventional as well as the marker-assisted backcross method. By integrating all these, we have developed this germplasm, and now we are developing hybrids also. So in this case, we have developed lines. Usually, lysine tryptophan is having negative correlation with methionine content. But we have break down that also, and we have developed few lines with high lysine tryptophan as well as high methionine content also. And I'm happy to share with you people that I have registered the last one at serial number 252311, that is the inbred line. We have registered first time in the world. This is the first inbred line which has been registered for high methionine content at NBPGR. And the first 4 also I have submitted, and soon I have talked to the head of department at NBPGR. They have told that during March there will be meeting and these lines will be registered. So we have made this line available for use. That will be available in public domain. This is the first line which we have registered. For improving the performance of inbred lines, in breeding session, I have shown that how US after 1944, they have stopped to use single cross hybrids and ask their breeders or their team to improve the inbred line performance first, then make crosses for F1 crosses and use. So we are working on that line also. We are selecting those lines. We are developing such lines which are having very good per se performance, means yielding ability. For example, these lines are having more than 30 quintal per hectare, means 3 ton per hectare. In case of inbred line, it is too high. And when we will use these such lines for hybrid breeding, so the hybrid seed cost will be lower down because more seed will be produced. At female line, if line is productive, more hybrid seed will be there. Always we will harvest hybrid seed on female line only. So female line productivity is also improved by these methods. We have developed lines by using DH method, which I have explained earlier. And these DS-based material, we have made crosses with male parent, and what we have got, you can see the performance, 10 ton yield. It is not less, it is too high under field condition or during kharif season only. These hybrids are very productive. And another program by using backcross method, because in case of quality protein maize, the genetic base is not so diverse. So to diversify that base, we have used private hybrids as female, then crossed with by using quality protein maize line, then made another cross, 2 back crosses, then selection was made for less opaqueness with hard kernel and high yielding genotype. And now we have developed these lines also with very good per se performance, and they are giving very good combinations also. You can see this at kernel station. You can see the yield level more than 12 ton. So this is remarkable achievement in our breeding program. And in one slide in breeding program, I was discussing that how we are categorizing our material into 2 different groups. Some lines are behaving like male and some lines are as female. So this is the method, heterotic grouping. We are made cross with 2 tester. The line which is giving higher yield with tester 1 means female line. Female के साथ अच्छा combination there, it means हमारी line में male का trait है. That we will put as line B. Or the line which is giving higher yield with the tester 2 male parent, that is behaving like female. So that will go to A group. So like that we have divided our material into 2 group A and B, female and male. Now we will use A and B line to make the heterotic combinations to have higher yield. So we have developed very good hybrids, IQMS203. That is a very popular hybrid, grown in Rajasthan, Gujarat, Madhya Pradesh, Chhattisgarh area. It is giving very good yield, and this is tolerant to major insect pest also in that area. And I am happy to share that the state government has floated a tender for this hybrid, 20,000 quintal seed for coming Kharif season. The companies are running behind for this hybrid. Pusa HM4. HM4 was released by HAU, and then our IRI colleagues, they have converted these hybrids through marker-assisted backcross method in QPM version. Again, Pusa HM8 also, like that only. HM9, then QPM with provitamin A. Hybrid, Vivek QPM, Vivek hybrid 9 was there earlier. Then it was converted to QPM version. One student was asking that how we can take challenge of converting one hybrid with multiple traits. So you can say this hybrid is having high lysine, high tryptophan, then provitamin A also. All these three traits are combined together in one hybrid. Similar with the situation with these also. So these are the some hybrids, and now we will move to phytate reduction program. This I have already explained that high phytic acid chelating with the availability of available iron, zinc, potassium, magnesium, calcium, all these micronutrients. And by reducing the phytate level, we will made available these micronutrients. So this is the method by which we have generated this you can say this low phytate mutant, and that mutant is used to produce or to lower down this phytic acid. And we have targeted 3 hybrids together, PMH1, PMH6, and 121. These all hybrids are popular hybrids grown by farmers or developed by different institutions, PAU Ludhiana, and one student was from PJTSU. The DH121 is developed by PJTSU. Then marker assisted selection by using that is gene linked marker. These markers are there, and we have employed this SNP marker, and then completed this process. We have developed nils and then estimated the phytate content, and using that material we have developed one hybrid, first hybrid of the country, which has reduced level of phytic acid, PMH1 LP. We have introgressed this LPA into PMH1. You can see we have reduced this phytic level from 2.94 to 1.89. This has been developed and released. Now this is the driving new germplasm for Maize or with the high amylopectin content, and we have targeted few private hybrids to develop the more diverse germplasm by using these hybrids. And Vexi pollen, we have used this from this Vexi germplasm, which we have developed earlier, and again targeted these much private hybrids, and according to 2 pools, in the heterotic pool 1, 2, and we have analyzed this for amylopectin. After introgression program, we achieved the level of more than 95% in both the cases. So we have done this successfully. And this slide is for how we can address the issue of yield penalty. So we have estimated that also. This experiment is under process now. And the QPM hybrids, which we have released earlier, they are also converted to this Vexi version. So this program is also successful. we have developed, and this foreground background selection that needs to be separate session. How we are doing this? It's a long process. We have completed this also. You can see this background recovery level is approximately 90%. In both the cases, this is the female parent, this is male parent. In both parents, we have achieved the background recovery level by using backcross method or marker assisted backcross method. After 2 backcrosses, 3 backcrosses, we achieved the 90% level. These are the lines which we have developed. So in brief, you can see this, we have done this, and through molecular breeding program, we have targeted different hybrids for which you have seen in last slide is high amylopectin, you can see for Vexi program. One disease we have considered high lysine tryptophan, provitamin, and low phytic acid, which we have complete right now at this stage, and we have released these hybrids. This is CMH 08282, 292. These 2 hybrids we have taken for this Vexi program, and these are the release for different zones, and we have completed this and contributed to the ICRP system for testing, and this is the yield level which we have achieved. These are the developed converted hybrids, you can say. From normal maize to Vexi version, we have converted this, and this is the level of their yield. Overall yield, these hybrids we have developed QPM hybrids through molecular breeding, and we have released all these. So in one slide, I was discussing about this quality. In case of quality protein maize, there is no reduction of lysine in tryptophan. This experiment I will explain by these slides. We have made chapatis of quality protein maize as well as non-QPM or normal maize. You can see the quality, there is no breakage, the cracks are there, less water is required when making dough with QPM, whereas more water was required because this is a coarse material, this is fine material. And color is, in case of this, is also acceptable, whereas this is yellowish color. People don't want yellow type chapatis. Yellow, they need only that chickpea chapati only. So in this case, shear force is required to break down the chapati. जब आप दांत से चपाती को चबा के खाते हैं तो आपको सॉफ्ट चपाती चाहिए, सॉफ्ट चाहिए, कम पावर लगनी चाहिए, ताकत कम लगनी चाहिए, आपके दांत जाड़ को परेशानी नहीं होनी चाहिए. That's why we conducted this experiment, and there is a machine that put force to break down that chapati, and that force is known as shear force. And the quality protein maize needs less force to break down that chapati as compared to the PMH10 or this MCFL 346, means white hybrid 574. These are the normal maize. These 2 are QPM and 1 is the landrace, desi makki. So QPM is as good as the desi makki. So less force is required to break down that chapati. It means the person who used to take chapati, made chapati, they like it. Again, the quality, lysine and tryptophan in case of flour, in case of QPM, and when we made chapati also, you can see the lysine, this is more than 4, tryptophan 1. So there is no reduction, almost no reduction in case of QPM, even after cooking, so intact quality. And overall, you can see the color is acceptable, taste and overall acceptability. The people have accepted the quality protein maize. They are having very good chapatis. We have conducted this survey by putting label with coded. So even I don't know the chapati is made of this QPM or normal maize, and served to number of peoples, and put one paper along with that sample that you make your preference, you give your marks, then averaged for color, aroma, taste, and overall you can see this is the acceptability. QPM is the most accepted one with highest score. We have made so many food products by using QPM, barfi, cookies, biscuits, chakli, phakli. We have made too many products. You can see this porridge, sattu, biscuit, kheer, halwa, upma, health mix, or supplement mix, khichdi, etc. for infant foods as infant foods, which can be provided to the growing children, and then preschool going children, age of more than 5. These children can take these items easily. So nowadays, our major population, they are loving the fast food. So we can make fast food with this also at our home. It is very easy to make, and other products, laddu, khazur, we can mix with QPM, we can prepare with QPM flour. So these are all are nutritional rich products which we can make with the UPM, and the young students, they can develop entrepreneurship also. We are giving trainings to them. We are providing some platform also through one program is there at Hyderabad, at Delhi, under ITMUs. We are providing some funds also so that they can develop their own business, and we are giving handholding also. So we have published literature also for production of these products. And one more thing, corn silage. That is the most important business right now. People used to have a farmer who is growing maize, who is earning 45 to 50,000 in only 80 days, 75 days by growing maize, and they are selling to the silage industry. And one person is having silage industry in maize area. He is purchasing standing crop. He is harvesting himself. You can see this first machine that is harvesting and directly putting into truck, and then truck will come to at one place and the bale will be made, and this is the bale, and this will be supplied to different parts of the country. So you will have the good health of the animal, and UPM or quality maize can play important role. Once, because the silage is made at later stage right now, when grain is almost mature, somewhat hardness is there, so lysine tryptophan has already been developed. So that rich product will be fed to your animal, the muscle mass will be more, health will be good. In lesser time, your calf will be ready for next generation. So this can play an important role. And with these words, I thank you all that you have taken interest to listen me patiently. And since last approximately 2 hours, you are listening. Thank you very much. If anyone have any query you want to ask, you can ask.
Thank
Ajay?
you very much, sir.
Yes. Any query? Time is too less for me because I have to travel tomorrow early in the morning. I have to made myself ready for that. Please. You
Thank
can
you
take
very much, sir, for your insightful words. Sir, we are extremely grateful for your valuable time. We will now be moving on to the query and interaction session. Before we begin, I would like to briefly share a few instructions for the participants. Those who have questions or doubts are kindly requested to send their queries to our team members on your behalf. We will ask the questions to our esteemed speaker. We request you to keep your questions short, clear, and relevant to today's topic. Participants only post their question in the chat box.
Dear students, if you don't have any question, we can wind up, and on another day I will be available with more breeding program, elaborated breeding program for 2 hours. Complete breeding we can discuss. Sorry.
Okay, sir.
थोड़ा सा मुझे आज
On,
मैं अकेला हूं घर में, मुझे खाना पीना भी देखना है, तो काइंडली एक्सक्यूज मी.
okay, sir. Thank you, sir. On the behalf of entire Agri SAARTHI team, I extend my heartfelt vote of thanks to everyone present here. First and foremost, I express our deepest gratitude to Dr. Ramesh Kumar, Principal Scientist, Genetics and Plant Breeding and Maize Breeder, for delivering such an enlightening, impactful lecture on biofortification and maize breeding. Sir, your insight into improving nutritional quality through scientific maize breeding have truly broadened our understanding of how much agriculture can directly contribute to combating malnutrition and ensuring food security. And it is rightly said, agriculture is the foundation of civilization and innovation in agriculture is the key to a healthier future. And today's session beautifully reflects this thought. Your experience, practical examples, and research perspectives have inspired us to think beyond conventional farming and move toward nutrition. We are also immensely thankful to our respected President Dr. O.P. Chaudhary sir for his constant motivation and guidance. A special thanks to our Chief Secretary Ajay Kasana sir, whose dedicated coordination and leadership ensured the smooth execution of every event. Your effort behind the scenes truly make this program successful. I would like to extend gratitude to all faculty members and students for their enthusiastic participation. Your curiosity and engagement made the session even more interactive and fruitful. Now, let me conclude with a beautiful quote. The future of farming lies not just in feeding people, but in nourishing them. Once again, thank you, Dr. Ramesh Kumar sir, and thanks to everyone who are contributing to making this session grateful. Before concluding, I would like to remind all the participants that the attendance link has been shared in the comment box and this link will be active for 30 minutes. Kindly mark your attendance. Thank you, sir.
Thank you. Thank you very much for providing me an opportunity to meet all these young minds. And perhaps I have explained only 1% to them. On another day, we'll try to have more information for you. Thank you. Thank you, Ajay.
Thank you, Dr. Saab, and thank you to all the participants.
चलो भाई ठीक है. Okay, close the meeting.